Hey guys, welcome back. Today we'll speak about glycogen metabolism, glycogen storage diseases, and regulation. So, blood glucose can be obtained from three primary sources. We have the diet, which we already spoke about in my last video, glycolysis. Then you have degradation of glycogen and gluconeogenesis. And in this video, we are going to speak about glycogen because I've already made videos on the diet and gluconeogenesis. In the absence of a dietary source of glucose, it is rapidly released from glycogen stores. So the main stores of glycogen in the body are skeletal muscle and liver. The function of muscle glycogen is to serve as a fuel reserve for the synthesis of ATP during muscle contraction. And the liver glycogen, the function is to maintain the blood glucose concentration. Now let's move on to the structure of glycogen. So it is a branched chain polysaccharide made exclusively from alpha D, which is dextroglucose. Now the primary glycosidic bond is an alpha 1,4, first carbon of one monomer and the fourth carbon of the second monosaccharide linkage. So alpha 1,4 linkage. There are also branches, so it's a branch structure containing an alpha 1,6 linkage. 1,6 means it's going up, not horizontally, so it is branched. Okay, let's speak about glycogenesis, production of glycogen. So the process occurs in the cytosol and it requires energy, which is supplied by our favorite ATP for the phosphorylation of glucose and uridine triphosphate, UTP. Okay, so a protein called glycogenin, it can serve as an acceptor of the glucose residues from UDP glucose. Now, glucose is converted to glucose 6-phosphate in glycolysis, and this is converted to glucose 1-phosphate via phosphoglucomutase, which then attaches it to UDP. So there is a UDP glucose, which gives us the glucose with the help of the protein glycogenin. Now the reaction is catalyzed by glycogenin itself via autoglycosylation. This means that glycogenin is also an enzyme. This short chain serves as a primer, a starting point that is able to be elongated by an enzyme called glycogen synthase. Now, glycogen synthase is responsible for making the alpha 1,4 linkages in glycogen, the straight horizontal branches. It only elongates the already existing chains of glucose. The branches are made by the action of another enzyme known as the branching enzyme, which functions as a 4,6 transferase. Moving on to glycogenolysis, the breakdown of glycogen. So the branches, they are removed by two enzymic activities of a single bifunctional protein, simply known as the debranching enzyme. Glycogen phosphorylase, it sequentially cleaves the alpha 1,4 glycosidic bonds between the glucosyl residues. Glucose 1-phosphate, which is produced by glycogen phosphorylase, is converted in the cytosol to glucose 6-phosphate by phosphoglucomutase. Alright, moving on to regulations. So the hepatocytes, they release glycogen-derived glucose into the blood to help maintain blood glucose levels until the gluconeogenic pathway is actively producing glucose. So it's not the primary pathway. Now, liver glycogen stores, they increase during the well-fed state and are depleted during a fast. So glycogenesis accelerates during periods when the body has been well-fed, whereas glycogenolysis accelerates during periods of fasting. Muscle glycogen is not affected by short periods of fasting and is only moderately increased in prolonged fasting, which is like weeks. In skeletal muscle, glycogenolysis occurs during active exercise and glycogenesis begins as soon as the muscle is again at rest. All right, let's review what we just learned. So in the liver and our muscles, glycogenesis goes from glucose 1-phosphate to glycogen with the enzyme glycogen synthase, and glycogenolysis goes from glycogen to glucose 1-phosphate through the enzyme glycogen phosphorylase. All right, now I want you to remember 3 hormones. You have insulin, glucagon, and epinephrine. Now, glucagon and epinephrine, they obviously increase the blood glucose level, so they increase glycogenolysis to receive glucose, and insulin increases glycogenesis to reduce the glucose and store it as glycogen. So, the binding of hormones such as glucagon to plasma membrane G protein coupled receptors, GPCR, it signals the need for glycogen to be degraded, either to elevate the blood glucose levels or to provide energy for exercising muscles. This results in the G protein mediated activation of adenyl cyclase. This enzyme catalyzes the synthesis of CAMP, which activates CAMP dependent protein kinase A, PKA. Alright, so we have 2 forms, an active form, which is A form, and an inactive form, which is B form. Now we have some enzymes. So starting with phosphorylase kinase, it exists in active A and inactive B. Now the active PKA, protein kinase A, phosphorylates the inactive B of phosphorylase kinase into the active A. And active phosphorylase kinase phosphorylates glycogen phosphorylase, which is our second enzyme with the same forms, to its active A form from inactive B, which then begins glycogenolysis. So what did we understand from this? That the enzyme protein kinase A, it phosphorylates phosphorylase kinase, which in turn phosphorylates glycogen phosphorylase, which produces glucose from glycogenolysis. Now, a third enzyme is glycogen synthase, which helps in glycogenesis. If you want to make the enzyme active, you have to dephosphorylate it. And if you want to make the enzyme inactive, you have to phosphorylate it. Okay, now let's look at allosteric regulation. So in the well-fed state, glycogen synthase B in both liver and muscle is allosterically activated by glucose 6-phosphate, which is present in high concentrations. Now, in contrast, glycogen phosphorylase A is allosterically inhibited by glucose 6-phosphate as well as by ATP. So synthesis of glycogen is activated by glucose 6-phosphate and degradation is inhibited by glucose 6-phosphate. Now also, phosphorylase kinase, the B form, can be activated by calcium calmodulin complex. Okay, and now going to muscle, glycogen phosphorylase, it is active in the presence of high AMP concentrations. The AMP, it binds to the glycogen phosphorylase B form, causing its activation. Alright, now storage diseases, glycogen storage diseases. These are a group of genetic diseases that result from a defect in an enzyme required for glycogen synthesis or degradation. Now, they result either in the formation of glycogen that has an abnormal structure or in the accumulation of excessive amounts in specific tissues as a result of impaired degradation. So a particular enzyme may be defective in a single tissue. If it's in the liver, it can give you hypoglycemia, or a muscle, which can produce muscle weakness. So we have 5 types of glycogen storage diseases. You can see the enzyme defects, the different enzymes involved, glucose 6-phosphatase deficiency and what it presents with, debranching enzyme deficiency, branching enzyme deficiency, muscle phosphorylase deficiency, etc. Okay, now last part of this video is about regulation of carbohydrates in general. So when you have high blood sugar, which is glucose, it promotes insulin release from the pancreas, which converts glucose into glycogen. So it increases glycogenesis. And when you have low blood sugar, It stimulates glucagon released from the pancreas, which stimulates glycogenolysis. Alright guys, that was it for this video. I'll see you in my next one where we'll speak about simple sugars like lactose and fructose. Until then, take care. Bye.